like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. And I'm right where I belong in my happy place. And I'm right where I belong in my happy place.
Welcome to the National University of Singapore, Yongloo Lin School of Medicine Healthy Longevity Webinar Series. It's an interesting night. We've got David Sinclair. I'm sure all of you are excited to hear what he has to say. And we'll be talking to him in a few minutes. And if you're going to have a lot of questions, so use the Q&A function. And we have Max here to jump in and ask questions. So that should be exciting. Before we get on to the discussion with David, we've asked Shiva Ravintharan in my lab to talk about a recent paper, which will be on rethinking old drugs and natural products for aging and age-related disease.
Hi, as mentioned, my name is Shiva and I am an RA in BK Lab, and today I will be presenting an ongoing project in the lab. This is a mouse intervention study that will take place over the span of 6 months in order to measure the health span of the mouse when introduced to certain compounds. In total, we will be looking at 4 different compounds at different dosages. The aim of the study is to create a platform for preclinical development of small molecules to target aging and allow translation into human clinical studies. This is the study design. Before the 6 months begin, we will be conducting behavioral tests to obtain a baseline of the mice behavior. Following that, we will be conducting the frailty test bi-monthly, and at the end of 6 months, mice will be humanely sacrificed for further research. The behavioral tests that we will be conducting include grip strength, rotor rod, Barnes maze, and the novel object recognition test. The grip strength test measures the muscle strength of the mouse limbs. This is assessed as they hold onto a bar or grid that is attached to a sensor. The rotor rod test evaluates the motor coordination and learning of the mice. Older mice have been shown to have impairments in both the grip strength test and rotor rod test as they age. Barnes maze is a test that consists of a circular platform with 40 holes on the edges, with only one hole leading to an escape tunnel. Mice are familiarized with the location of the escape tunnel with training and visual cues, and then undergo a probe trial where the latency and distance traveled before reaching the previous escape tunnel is recorded. Performance on the Barnes maze is linked to the integrity of the hippocampus, which is key for spatial learning. There are 3 strategies which mice can take in order to find the escape tunnel. Mice with less deficits to the hippocampus will be more likely to use visual cues placed around the platform to find the escape tunnel, while mice with deficits to the hippocampus are more likely to either find the escape tunnel randomly or in a serial manner moving along the edges of the platform, despite the previous training, indicating an impairment in the spatial memory of the mice. Novel object recognition is a test to investigate the memory of a mouse. It begins with training, where the mouse is presented with 2 similar objects and allowed to explore them. During the test, one of the objects is replaced with a novel object. Objects used should be easily differentiated and not too much larger than the mice. If a mouse is able to remember the familiar object, it would spend more time exploring the novel object. Long and short-term memory can also be tested by varying the time period between the training stage and the testing stage. Mice with less deficits in memory would ultimately spend more time exploring the new object. If a mouse has deficits in its memory, it would be more likely to explore the object that it should be familiar with. These tests would allow us to assess the effects that the different compounds and dosages have on the muscle strength, learning, and memory of the aging mice after 6 months of treatment. Other than the behavioral tests, we will also be using a 31-item frailty test. This test evaluates the frailty of the mouse across different systems. Scores are given a 0, 0.5, or 1, with increasing values indicating increasing severity in a deficit. Some of the 31 items include body weight, body temperature, loss of fur and fur color, and hearing loss. A baseline is recorded at 18 months with later evaluations compared against the baseline. Previous research has shown that aged mice have a higher frailty index score, indicating more accumulation of deficits. The frailty test would allow us to assess the effects of the various compounds on the frailty and health span of aging mice. With this project, we hope to be able to gather more information on which compounds could help extend health span and eventually bring it forward for clinical testing. Thank you.
Thanks a lot. And actually, I misspoke. That's like we've switched over to a new approach at the beginning of the show where we're actually having people at NUS talk about their projects. And so we've been testing a number of natural products on aging, and we're excited to see how some of these do as we look for continued ways to find small molecules to impact lifespan. And that leads in well to David Sinclair, who's another faculty member in the aging field, has made huge progress in trying to identify ways to extend health span and lifespan preclinically and I think increasingly clinically now. He's a tenured professor of genetics at the Blavatnik Institute at Harvard Medical School, a co-director of the Glenn Center for the Biology of Aging Research. And you have a few things, David, on your list that most people don't have. Time 100's Most Influential People. I haven't read that before in an introduction. And also an officer of the Order of Australia. So you have to tell us what that is. So I want to bring David on to give a short talk, and then we're going to do this mainly as a fireside chat tonight. So David, thanks for joining the show, especially at 7 a.m.
Oh, hi, Brian. Thank you very much for having me on. It's good to see you. I don't know if everybody knows this, but my career started with Brian. Brian taught me how to wrangle a yeast cell and figure out how old it was. So thank you, Brian, for that start to my career. And it's great to be here. My background is Australian. I was given what the equivalent would be of a knighthood from Australia, which was kind of them. And then I guess Time Magazine just liked the idea that we were slowing down aspects of aging by turning on longevity genes, which I'll talk about. Right, so I'll switch to my presentation. It's only a short one. Today I'm going to talk a little bit about the work that we've done over the years, looking at the role of epigenetics in aging, which really actually started with Brian as I came to the US from Australia. One of the reasons that I chose to study aging is that it could have a huge impact on human health. Most diseases on the planet now are caused by aging. We call them diseases, but heart disease, frailty, Alzheimer's, diabetes, these really are driven by aging. And we've made some progress extending lifespan and making people live longer. As you can see here, a lot of people can expect to live beyond 80. But we can do better because the current approach to medicine is that we put a Band-Aid on at the end of the aging process instead of intervening before it actually occurs. And there's some evidence from studying animals and looking at people who take certain drugs like metformin that we can protect people even further. Of course, doing the lifestyle changes that we all know are helpful, such as eating less often as well, and also exercise. And so eventually, I believe that we'll be able to have a world where people can live a lot longer and then die relatively quickly in their population. So I want to talk about the epigenome. I think many of you will have heard of this word before. The epigenome are the structures and systems that control the expression of genes in the genome. And it's essential for life, just as the genome is. And it also includes information. The epigenome is information that tells the cell which gene should be on, when, and where in the body. And without the epigenome, cells would all be the same because they all have identical or near identical genomes. And during development, cells lay down various mechanisms or structures that tell a cell whether it should be a skin cell or a brain cell. One of the structures or one of the chemical additions is a methyl, which is just a carbon and 3 hydrogens. You see here, methyl here on a packing protein called a histone. And over here we have a methyl, which is coming out of DNA, which is also very important for telling a gene, typically whether it should be shut down over time. And when I started in Lenny Guarenti's lab at MIT, which I can see out my window here, I'm here in Boston, what we did was, Brian discovered there was a gene that was involved in silencing genes. So the gene was called silent information regulator, and the I is information. And so what's come from that work over the last 25 years is the appreciation that the loss of information, in particular epigenetic information, is a key driver of the aging process, not just in yeast, but in mammals as well. And we can read these chemicals quite easily on a sequencing machine. And if you plot those changes and use some machine learning, you can find DNA methylation sites on the human genome. In this case, this would be a blood test, but it can be a skin, it can really be any part of the body. We find that it correlates with your biological age and can even predict when you're going to die. And this is primarily the work of Stephen Horvath, who gave us the name, the Horvath clock. And so there are a number of companies now that measure your biological age, your DNA methylation, and can tell you if generally you're older than the population or younger than the population for your age. And we also know, in large part from Steve's work, but now many others, that if you look after yourself, and 80 to 90% of how you live your life determines your future health, you will be younger if you've done the right things. If you exercised, you ate less frequently. These things tend to make people have a younger epigenetic biological age. So we decided to find a couple of things. One is what drives this forwards, what's causing aging to go forwards in the first place. And then, is there a reset button? Is there a backup copy of youthful epigenetic information in the cell that we could use to take somebody like this and reset them to be like this, and perhaps even like this one day on the left? So one of the main causes of information loss, epigenetic information loss in a yeast cell, was a broken chromosome, broken DNA. And we published this in 1999. And in 2009, we published a paper that said this is probably true for mammals as well, that broken chromosomes distract the epigenetic regulators, such as the sirtuins, which are not just in yeast, but are also in mammals. We have 7 of these sirtuin genes. And by distracting them, they actually can no longer regulate genes and protect the body against aging. And this mouse is the control mouse in this experiment, but we also have a mouse who's the same age, 16 months of age, where at 5 months of age, we disrupted its epigenome by creating 3 times the average or daily DNA break level. Now, importantly, these mice don't experience tumors, they don't get mutations above background, but it's striking what happens when you accelerate their epigenetic age. As you can see here, this mouse looks old, but it is literally older based on the epigenetic clock by about 50%. And it also experiences aging. This mouse has diseases of aging at the histological, biological, and of course, epigenetic level. We can also use molecules to reverse aspects of aging. This is a study of a molecule called NMN, which the body uses to make NAD. NAD is a chemical we need for life and goes down as we get older. But NAD is particularly interesting because it was discovered, again, in Lenny Guarenti's lab, that NAD is required by the sirtuins for their activity. And so without high levels of NAD, the body is not protected, the epigenome destabilizes, and you get old age. By giving the mice NMN, we could reverse that effect, and the muscles became more oxygenated and had more mitochondrial derived energy. And the mouse on the left clearly is the one that has been drinking NMN water, and the one on the right is just a regular old mouse. These mice are 20 months old, so this is the equivalent of about a 65, 70-year-old human. And if you're wondering, it only takes 4 weeks to get the increase in endurance by the NMN. And we know that this is dependent on SIRT1, one of the sirtuin genes, because we deleted it from the endothelium, the lining of the blood cells, and we did not get the effect. So what about the reset button? So we worked for over a decade to find a way to reset the epigenome. It's quite difficult because if you do it too much, you end up with a tumor, because a cell that has 0 age is not going to be healthy or have an identity. But we came upon a set of 3 genes that are in part used for, well, these and other genes are used to make iPSCs, induced pluripotent stem cells. And so Yamanaka, who won the Nobel Prize in 2016, used a set of 4 genes to create iPSCs, the O, S, K, and M. And we tried O, S, K, and M, and we tried to follow Juan Carlos Belmonte's work at the Salk Institute. And we kept killing mice, we kept causing tumors, and it was not looking very promising. Wencheng Liu, a student in the lab, decided to use O, S, and K and leave off the CMYC, the M. And it worked. We could reverse the age of human cells. They didn't become tumorigenic. And we decided to put it into a mouse. And we chose the eye because the eye is potentially a tissue that we could treat in humans with gene therapy. And now this is a mouse that was untreated. This is a 1-year-old mouse that is blind to these moving lines. Its eyes are not functioning and it cannot see. And we know this because its head is not turning to the right. But if we use this O, S, and K gene therapy to reset the age of the neurons in the eye, we literally restored their vision on average back to youthful levels. And we could see that the age of the neurons went back, and the gene expression and the epigenome also went backwards by about 50 to 75%. And that, what we call DNA methylation pattern, was reset largely by this treatment. And in this case, we let it go for 4 weeks. If we go for 8 weeks, their vision gets even better. And it's a permanent reset. We now have mice that die when they're old, but their eyes are still young, working perfectly. So we're excited about the idea of reprogramming an entire animal and, of course, eventually an entire person. We are looking at human clinical trials starting next year. We're in non-human primates right now for treating glaucoma, which is a leading cause of blindness. It does show, though, that there is a backup copy of youthful epigenetic information that resides in the cell somewhere, and we're looking for where that and how that information is stored. I wrote a book about this discovery and the work that I did with Brian. Brian is in the book, and you can certainly read more about it. And I talk about new papers and my thoughts pretty much daily on these sites on social media. So I want to leave it there for questions. I have a lot of people to thank. The 2 main authors of this paper are Jae-Yun Yang and Matoshi Hayano. And I also want to mention with the reprogramming work, Wencheng Liu and Xiao Tian. Thank you very much. I'll leave it here. Take questions, and we can also have a chat, Brian.
Sounds good. It's good to see you again. You know, this pandemic's keeping everybody from seeing each other, so we haven't been able to connect in a while. So nice to have a chance to talk. And Max sent me some pictures from our time in Lenny's lab, and I promise I won't show them. But it reminded me how long ago that's been and how many things have changed. So it was a great time in Lenny's lab, though. I think that there was a lot of ideas, and I think, you know, Lenny really helped us out a lot in terms of getting this aging project started. So I should have him on the show at some point. What do you remember from those days?
Gee, I remember a fair bit of alcohol. I remember sleepless nights. We were crazy. We would stay up till 1 o'clock every night, and that's only because we would have stayed later, but the trains stopped at 1. I do remember it being extremely competitive. We were all out to change the world. We thought that we were onto something that would end up really changing the world, and we're on that path still, which is really exciting. Yeah, but I remember there also being a camaraderie between us because we knew it was special, a very special time.
It was really a time of ideas. Go ahead.
Yeah, it was. The one thing you'll appreciate, but probably most people don't, is how unexpected it was to find a silencing gene involved in aging. It made no sense at the time. And your brilliant idea that it was controlling an age locus, a region of the genome that was involved in aging, really was the breakthrough that I think the field needed. But it took 20 years for people to get used to this idea and move away from things like antioxidant defenses and think about the epigenome.
Well, you know, we were working in yeast cells at the time too, and I think a lot of people, including myself, weren't sure that what we learned from yeast was going to translate to humans. And you know, I know now, you know, seems to be less and less focus on yeast. Do you think there's a future for model organisms like yeast cells?
Yeah, I definitely do. It's a shame that people have moved on, and that's partly because of what gets funded and fashion in journals and the NIH. But we still use yeast in my lab. We're looking at finding epigenetic stabilizers, and we have a gene called SSB1, not D1. And when we overexpress it, it extends the lifespan of yeast and prevents the sirtuins from moving too much. And it works in worms as well and in human cells. So, you know, the yeast definitely still have a role, and they've been neglected, but I think more labs should consider using them because the lifespan of a yeast cell is only, what, a week at most. And so, yeah, I'm still bullish on those little fungi.
Yeah, and also now there's robotic ways to look at the age of yeast, so we don't have to ruin our eyes dissecting all the time.
Right. And actually, when you started, Brian, you didn't even put them in the fridge. You would just go on shift work to manipulate them until they died.
Yeah, Nick Austriaco and I would sit there in the 30-degree room, and which was full of yeast cells, and we'd come out smelling like, you know, yeast, and that was for 2 months, I almost couldn't go home. But and Nick became a priest. I don't know if that had anything to do with it, but it's, we'll have to have him on the show sometime. You know, let's talk about aging a little bit. You know, I noticed you mentioned in your graph that you know, things are good. You kind of pushed out the curve, but you still had a clear maximum on lifespan. So, you know, we talk about slowing aging, we talk about reversing aging, we have people thinking pragmatically and people thinking futuristically. Where are you on this spectrum?
Yeah, I'm definitely on the spectrum in many ways. I'll speak practically because there's really two answers here. Practically, it's easy to extend your lifespan. This is the main thing that I try to get the message across, which is that if you do the right things, which is don't overeat, eat less often during the day, you do some exercise, you don't smoke, don't overdrink, that alone gives you, compared to people who don't do that, 14 extra years. So living longer isn't hard, it just takes some discipline. And that could already push the curve out to the right by many years. In addition to that, we have already supplements that really do need better regulation, but I believe can add some extra years to life. Brian, your work I include in that, great work on alpha-ketoglutarate. And then we have medicines that are seemingly helpful based on studies of tens of thousands of people. Metformin is the most well-known and perhaps the safest of all, which could extend life another few years if more people took it. That seems to protect those at risk against heart disease, cancer, Alzheimer's, frailty, and of course, type 2 diabetes. And it's increasingly being used across the world as a preventative measure as well. So I think giving the average person an extra 10 years is easy if people just do those things. But what about, let's look into the future. How far out can we push it? Can we all expect to live beyond 100, or most of us? I think that it's doable within our lifetime. The technologies that I'm seeing being developed around the world and the amount of investment, which is in the tens of billions of dollars now, will lead to breakthroughs we can barely imagine now. And the longer you live, the better chances there are that you'll live longer, because the technology is catching up so fast. Is there a limit to human lifespan? I don't know of any theoretical reason why we have to die. There's no program that says once you get to 120, you drop dead. But it gets harder and harder the further out you go. So I think that this talk of living to 200, I don't think there's any way in our lifetimes that we're going to get there, unless reprogramming turns out to be a wild success and we can reset the body multiple times. Again, never say never, but there's a long way to go.
Well, I think the reprogramming is one of the possible ways to really have a huge impact, and your study is one example of that. There have been other studies suggesting reprogramming may have impacts on aging too, with the minor cancer caveat. I think that some of these new areas have the potential to lead to huge breakthroughs. The nutrient signaling stuff, I think, is going to cross the first thresholds to have a real impact, but I'm not sure it's going to get much beyond that. I think that if we're really talking about big change, we need something like reprogramming, gene therapy, cell replacement. I don't know which of these are going to work best, but it seems like the reprogramming stuff is getting more and more exciting at the moment.
Well, I would agree with that. The ability to really turn back the clock. And what's fascinating are the studies that are showing that you can reverse the epigenetic age of humans.
Yeah.
Admittedly, it's only a couple of years now, but I was at a conference a couple of weeks ago where people are claiming have gone back multiple times. The people there that say that their blood's epigenetic age is 10 years younger than it was a year ago. And then it gets really interesting. If you can truly take your age back more than a year every year, then theoretically you're immortal. I don't think we're there yet, but we are approaching a time where what was seemingly impossible just a few years ago is now able to be achieved even in humans.
I'm a proponent of these biologic clocks. I think they're really, we talk about interventions as changing the world in aging research, but being able to measure the rate of aging is equally important. How much faith do you put in these clocks? I think at this stage, there's a bunch of them. There's a lot of questions that come up. Is it something specific about methylation, or can we measure it from any complex data set? Is it really aligning with biologic age, or just telling us something about aging? And that's important because, if you're reversing the clock, are you reversing aspects of aging, or are you really reversing your age? And I think those are questions we're trying to figure out in the field right now.
Right. Well, there are lots of clocks. There's epigenetic clocks, glycan age, immune age, there's proteomes. There's just blood tests looking at 40-something parameters. You can make some estimation. I tend to like the clock that says that I'm the youngest. No, but in all seriousness, I think the compendium of all of those is going to be the most useful. There's no one clock to rule them all. But if I could only pick one, I would go for the epigenetic clock, because it seems to really be stable. It's not going to change based on what you drink that day. And also, we had that Nature paper in December about the reprogramming, and we actually found that DNA demethylation was necessary for the restoration of function in the eye, arguing that this process of, or at least what the clock represents, may be part of the aging process itself, which is a profound thought if it's true. So I think that the epigenetic clock is important, but I also think it's important to measure the physiological changes as well. because those are clearly an important part of day-to-day life and health. The one that I take, and I've been taking or doing for over a decade, is a blood test looking at biochemical parameters, a company called Insight Tracker where I consult. And so that's been very useful. And if nothing else, what it does is it motivates you to change your lifestyle when things start to go in the wrong direction. And that's also an aspect that I think is going to be very important in the future, over the next 5-10 years, giving people a number to focus on. Otherwise, really with your own wellness and health, you're flying blind.
Yeah, and I think that one of the major things is to get people to give people tools so they can really take charge of their health. It's easy to tell everybody to exercise and eat right. People don't want to do that. I think if you have numbers they can get, tests they can do, at least for some people, that's really going to motivate them to try to optimize their health. So I'm excited about some of these approaches. I think it's more and more the public is paying attention to this too. I'm sure you get contacted a lot by people trying to measure their age, but it seems like it's really catching on. It's been a sort of a long-term PR attempt. We've both been involved in that, and other people in the aging field as well. Do you think it's finally mainstream now, trying to change your age?
Well, in my circles, it is. People that I talk to in the public or in the news, or people who've read my book are all into this. It seems to be penetrating the entire planet. I would call it a mega trend. And even at the highest levels in government, and those who have a fair amount of money to spend are really paying attention to this field now that it looks like the science has caught up to where we want it to be. Similar to where the cancer field was in the 1970s and 80s with the discovery of oncogenes. The field of aging now really has matured to a point where some more effort will have a dramatic, and money will have a dramatic effect on people's lives.
Yeah, and there's a lot more money now. I mean, when you started, was involved with starting Sirtuins, I mean, there was Sirtuins and Elixir and maybe Mike West and a couple other people doing stuff, but really, it was the private sector wasn't interested in aging, and now, especially in the last 5 to 10 years, it's a crazy explosion of interest. Maybe you can comment on that a little bit.
Yeah, right. Well, when Sirtuins was started, it was a very strange thing. Well, people thought it was strange to work on aging. They didn't understand the concept. Why would you work on aging? It's not a disease. It's not treatable. Well, the goal was to show that you could use the discoveries out of the Sirtuin field to treat diseases. And we had positive data in psoriasis, the skin inflammation disease. Ultimately, those drugs were not pursued, actually, not for scientific reasons, but for business reasons. And we've revived. There are now at least 3 companies working on Sirtuin activators, including actually one that I'm working on with Lenny. So that vision isn't dead. But it was strange in those days, and now it's really mainstream. I was at a conference a few days ago, packed with people wanting to invest in this area. And there's at least $20 billion with a B, US dollars, that has been already gathered to invest in this area. And the pace of investment, the pace of research is making my head spin. It's very hard to keep up with all that's going on now.
Yeah, I agree with that. Of course, when we declared war on cancer, I think a lot of people would have said we would have cured cancer by now, 50 years later. And it's certainly a lot of progress has been made, but we haven't cured cancer. So do you think aging is going to go faster or slower, or it's going to be, what are the hurdles along the way, and how long is it going to take to make real impact in this field? And I realize that people can exercise and do stuff now. I agree with you on that, but to really change the world in terms of targeting aging, what's the time frame, you think?
Yeah, well, we're not going to have a pill that stops aging anytime soon. So there will be advances that will together have a big impact, similar to cancer, where we have chemotherapy and then we add immuno-oncology therapies. And we have made a lot of progress. There's a lot of people who are alive today that would have died from cancer 20 years ago. And so it's going to be the same. We're not going to all suddenly live 100 years longer, but we are going to slowly live longer and longer and longer. But I think there are two differences. One is that treating aging seems to be much easier than treating cancer. I wrote that in my book, and a lot of people were surprised by that. But while cancer is literally hundreds of different diseases, the main drivers of aging are common across the body, and the protectors against aging, such as the sirtuins, are common in tissues. So if we can target those, I think we have a good chance of going faster than cancer. And then the other reason is that with aging, we actually don't have the issue where we have to rely on 20th century technology. We can do 1 million experiments today that would take a decade back in the 1970s. Mm-hmm. And so the ability to develop drugs and find new ways to treat aging is exponentially faster than it used to be.
Yeah, and I think I agree with you on the idea that treating aging is going to be easier. I think that as a preventative approach, it's really more about maintaining homeostasis and possibly reprogramming events that are happening during the aging process. Whereas, cancer is a state of disequilibrium. It's really a challenge to reverse that. And I think that if you catch things before they go wrong, it's going to be easier than waiting until they go wrong. Maybe we can talk a little bit about sirtuins. I think that you've worked a lot on SIRT1, and you're probably still very bullish on the idea of SIRT1 regulating aging. There's 7 sirtuins, so maybe we can start broadly though. And which are the other sirtuins that excite you, and why would this class of proteins regulate aging?
Well, so there are 7 of them. There are 3 enzymes in the nucleus, and there are 3 in mitochondria, 1 that's cytoplasmic. And they play protective roles throughout the cell. There's now thousands of papers since Brian's first one that show that sirtuins protect cells and protect tissues against the ravages of time and aging. And there are a number of studies that show they affect the aging process as well. The one that seems to be the most potent, or perhaps the easiest to use in an animal, is SIRT6. SIRT6 is similar to SIRT1 in that it binds to chromatin and regulates genes, and also is involved in DNA repair, broken DNA repair. And what's been striking is that Chaim Cohen, a former postdoc in my lab, has shown now in a number of different ways that SIRT6 is a longevity gene. And when you make a mouse that overexpresses it, it will be healthier and lives longer and is resistant against diseases such as type 2 diabetes. Now, what's exciting about that is that you can activate SIRT6 just like you can activate SIRT1 with small molecules. And that's what these companies are doing, is to find a drug that could activate SIRT6 to treat a variety of diseases. Cancer is one possibility. But also it could be one of the first molecules to truly slow down the aging process.
Yeah, on question of SIRT6, how much of a role is NAD playing in SIRT6? NAD levels go down with age, as you mentioned. They're activators of sirtuins, and there's been a lot of data on SIRT1. I haven't seen as much data on whether NAD is a rate-limiting component for SIRT6 or not. Is there good data on that? Do you know?
Yeah, you're right about that. We've done a lot of work on SIRT1 and NAD's role, right? I mentioned earlier, raise NAD in the endothelium and block SIRT1. I think it's just because of lab's preference, not because of biology. Chaim, who works on SIRT6, and Raul Mostoslavsky and Vera Gorbanova, they don't think about NAD. But I would be surprised if it's not just as important for SIRT6 as it is for SIRT1. It's just that the experiments haven't been done yet.
Yeah, I think those are critical experiments to do, because I agree that SIRT6, I think, is a really strong candidate among the sirtuins to target for aging. I guess, we have to ask the question, what's the best way to raise NAD? Is it, I know you're a proponent of NMN. There's been a lot of talk out there these days between NMN and nicotinamide riboside. And what are your thoughts? I think I know what your thoughts are, but it'd be great for everybody else to hear.
Yeah, well, I don't sell anything on the internet, so I don't really care which is better at this point. Some professors do sell stuff, and they're often out there claiming that NR is better than NMN. NR is used by the body to make NMN, by the way. I do go on data, and in mouse experiments, and Matt Kaberlein, our colleague, agrees that NMN has a more potent effect compared to NR head-to-head in some areas. No one's done a human clinical comparison of NR versus NMN to raise NAD. And so these arguments are really semantic. What I can tell you is that NMN and molecules like NMN work really well in people to raise NAD levels 2 to 3-fold in the blood, in white blood cells. And we're doing trials here at Harvard and around the country in the US to treat diseases. And so we're looking at one particular disease called Friedreich's ataxia. We're also looking at muscle strength, oxygenation, endurance. And so I'm hopeful that at least for NMN, we'll have some really clear and hopefully positive human data using NMN or molecules similar to it to treat a human condition. And those results will be coming out early next year. But I don't like getting into the debate about which molecule is better. There's a whole supplement war out there that to me is something I don't even want to get into. And I want to just see the data. And I wish people would do more head-to-head studies of these molecules rather than just theoretically try to predict which one's better or base it on which one they want to sell more of.
I think that's a limitation in the field, both on the academic side and on the private sector side, is that people are still of the approach that they're testing their molecule and trying to do things side by side and compare them is missing. I mean, that's one of the reasons we're trying to do this wide array of study of natural products and side by side and compare the benefits and potential problems with each of them. I think that I agree more of that needs to be done because it's easy to argue.
Brian, I forgot to mention, we have put NR and NMN head-to-head in those endurance, those treadmill studies. Mm-hmm. And NMN was superior at the same concentration for reasons we don't know, and I don't even need to know. But that's one important data point that based on that, NMN was working better. But you're right, even my lab could do better comparing those two molecules.
One last question, there's a lot of questions from the audience, so I want to give Max some time. You mentioned in one of your recent papers about targeting COVID-19, and actually we've both written reviews on this. And do you think that targeting aging is an interesting way to augment things like vaccines in terms of targeting COVID-19? And are there particular strategies in aging that might work best?
Well, I do, and I think you would agree that by far the biggest risk factor is age, like 10 times more than anything else. And by reversing or improving the age and the resilience of the body in old age, I think is the best approach besides vaccines to be able to deal with this and future pandemics. The reason is that as the body gets older, the immune system declines and overreacts often to an invading organism or virus. And we know from studies that if you regenerate or improve the health of the body, it can fight diseases. And there are a number of clinical trials using molecules that are thought to slow aspects of aging or reverse it. There's a trial with NMN, there's a trial with NR, metformin, and even rapamycin. And then there's another class, which we haven't brought up, called senolytics, which seem to also be important, particularly in treating long COVID, which may in fact be driven in part by those accumulation of senescent cells in the body. So yeah, I think that really a lot of focus should be put on treating aging itself as a way to allow older people to combat COVID-19 and other infectious diseases.
Yeah, I agree. I think it's an interesting topic, and we need to put more emphasis on that because the vaccines are going to be a continuing battle, and there's got to be some strategies to help them work better. And then part of that's by targeting the body, as in addition to the virus. Max, Max Unfried has been a graduate student in my lab for a while now, and he's doing a sabbatical with Morten Scheibe-Knudsen. And Max, the more you spend in his lab, the more conservative and business-like you look. I don't know what that means, but can you comment on how Morten's affecting you?
I know he just has better style than you. I don't know. It's a Danish style, Those fashion icons.
There's a million questions, so you have to filter through these and pick the most interesting ones. So no risk here. So
absolutely, we also had some questions from Twitter, but I will divide them in between. Maybe one question to start with: when you guys were in the aging field and started it, or your PhD and your postdoc, you were quite rascals, because nobody else wanted to do it. What kind of advice would you give young researchers that would to enter the aging field now that it's becoming trendy and popular?
Brian, do you want to go first?
Sure, I'll take a shot at that. I think that one of the most important things is don't be conservative when you join a lab as a graduate student or a postdoc. You have to ask the big questions if you want the big answers. And, to me, I'd done experiments in yeast before, and I think you had too, David, when we came to Lenny's lab. I think what excited us is that this aging was a big, difficult question that we really had no idea what the answer was, and we felt like we had a handle on how to at least get some of the answers. And so it was a risky project. There's no doubt about that. I had a lot of people at MIT with very big names, some of which are working in aging research now, tell us that we were crazy at the work in aging, and we would never learn anything from a yeast cell. So you have to really identify an exciting question and not be afraid to go after it. That's the one thing I would say. David?
Yeah, one of the reasons I wrote the book was to just immortalize that. It's easy now to look back and say it was obvious, but we were criticized. Brian and I would give talks at MIT, and one professor I remember, who I will not name, said, "Oh, I can make yeast age. I just put them in the autoclave." And he totally missed the point of what we were doing. And it was disheartening, but we had the guts to do it. And the other thing I will tell you is every time we made a big discovery, I knew it was important because people would tell us that it could not be true. And that continued through my whole career. So have the guts to believe in yourself, do the best science you can, make sure it's right and reproducible, and then stand by it and just continue on. And if you have critics, it's actually a good thing, because I don't know anybody who's successful who doesn't have critics.
Yeah, I mean, David and I haven't even agreed all the time throughout our career. And I think that it's not just agreement, but it's also disagreement that drives the field forward. So you can't be afraid to pursue your ideas and defend them as well.
Cool. Let's get a little bit to the scientific questions. From Antonia, what is the mechanism that allows 3 of the 4 Yamanaka factors to reprogram the epigenome of eye cells?
Well, that's what we're working on right now. We are mapping the distribution of those transcription factors over time. Where do they go on the genome and what effect do they have? And what are the marks on the epigenome that change? There's clearly a cascade of events that happen over about a week or 2 weeks to reprogram the cells and establish a new 3-dimensional structure in the nucleus. How that happens, we really don't have a good idea. One big question is, where is that information stored? How does the system know where to put on a new methyl or subtract an old methyl? And we don't actually know that. We speculate there might be new types of DNA modifications or RNA or proteins involved. But yeah, this is really the big question. We do know that DNA demethylation, active enzymatic removal of a DNA methyl, is absolutely necessary for it to work. But what controls that and what downstream effects they have, we don't know. And I think this is one of the exciting parts of this new field that if any young person wants to get into, it would be a great question to try to address.
We have like a question from the Health and Wealth Twitter account, and they want to know, as a consumer, how do you evaluate which commercially available epigenetic test is the best? They all have like proprietary algorithms, which makes it hard to compare.
Yeah, that is a problem. And if you take two, you get different answers. I think it's just because it's the early days of really any product. It will all come out in the wash, as my mother would say. What I do is I've only taken one of these tests, but I think that I like to go with the one that has the best scientists and also the most detailed report. I'm even developing one of these tests. The reason is we've brought down the cost of the test by more than 100-fold. So it should be able to be affordable by everybody and be able to be done every few months. But yeah, it's a problem. But I think eventually we'll figure out a standardization. And I know Steve Horvath is working on making a standardized clock that we can all compare our clocks to.
Yeah, it's a problem, but it's a good problem. I mean, the real problem was 10 years ago when we had no way to measure anything. So I think this is just a hiccup along the way, but I'm excited where these clocks are going.
We have got a question from Wilbert, and it's like, do you think elevated levels of DNA breaks in your epigenome system influence aging of the mice, or can you disentangle both epigenomic and damage-driven effects?
Yeah, well, the damage is necessary, but it doesn't, in our system, it doesn't leave mutations. So we've sequenced deeply through these mice, and we can do this in cells. So it's not mutation-driven changes, it's epigenetic. We know that for a fact. And we also know that we can drive the age of the mouse forwards, but now we can also reverse it. And I mentioned Xiao Tian, he's from Vera Gorbanova's lab. Those mice actually get dementia, they lose their ability to learn, and then we can use OSK to reverse that process. And the fact that we can drive aging forwards and backwards is even more evidence that it's not due to mutations, it's due to epigenetic changes that are reversible. And also that we can stabilize the epigenome and extend lifespan is another good bit of evidence that it's not the mutations that are causing this. But we do need DNA damage signals to drive this. And a great experiment, which we haven't fully done yet, would be to create the DNA damage signal without DNA damage itself. And if I'm right, it should accelerate aging as well.
With regards to aging, Sarah has a question. Would you say that biological aging over the years is a linear process, or would it resemble more of an exponential process where there's a tipping point over? Well, based on the clocks that Steve and others have generated, it's linear, but I think that they aimed to find a linear correlation. Clearly, physiology declines exponentially because you can measure it, and then the tipping point is the point where an organ fails or a person dies or both. But yeah, early on it's pretty linear. You can graph epigenetic changes, and a lot of these changes in physiology, glucose levels change linearly. But towards the end, noise ensues. There's a lot of chaos, and systems break down very rapidly, and one problem leads to another. And in fact, the hallmarks of aging, of which there are at least 9, are interconnected. And when you start to break down in one, it does affect the others as well. But I'd be curious what Brian thinks too.
Well, I think that last point is the key thing, is that we talk about different pathways controlling aging. And really, it's a network that involves all of these hallmarks and pillars of aging. And that's good early on because it allows for compensation and integration between the different pathways. But when enough of them go wrong, it starts, one brings down the next, and it's a tipping point. So I would agree with that. So.
But if you would go for interventions, then would you try to have interventions before the tipping point or slightly after? Where would you target? Me,
I'll go first. The earlier the better. I was asked yesterday, how young should people try to improve their longevity? Now, I'm not saying take drugs and supplements in your 20s. I don't think we know yet if that's advisable. But in the mouse studies, the earlier you start them, the better, typically. Now, there are some exceptions. Reprogramming works late in life. Senolytics that kill senescent cells work late in life. But these nutrient modulators and NAD boosters, metformin, tend to work better the longer you do it for. And I think it makes perfect sense that you don't want the system to be chaotic and over that tipping point or anywhere near it before you start intervening.
Yeah, I completely agree. On a related note, what do you think about people taking supplements and drugs? I agree with you on 20-year-olds, but how about 50-year-olds? Is this the time when they can do that? I know a lot of people are taking supplements, and we both support that in various ways. And then, a lot of people I know are taking drugs off label too. What do you tell people when they ask you those questions?
Well, speaking as a 52-year-old who does that stuff, it's hard to deny it.
Yeah.
But I certainly don't ever recommend something. I'm not an MD, and we literally don't know the effects on individuals, especially in combination. We don't even know if you combine a few of these things, if it's good or bad. And that's an area that we need to know more about. Even in animals, we very rarely combine therapies to see the effects. But on the other hand, we know what's going to happen to us if we don't do anything. The end of life is not pretty. And so I've taken the individual decision to try things that are based on science that are not likely to hurt me. Resveratrol, NMN, metformin. And so far I'm still alive, but it's risky, right? Taking things like this is unproven. But consider the flip side, which is, in 30 years, if I don't do anything, I'm going to be probably frail and full of diseases. So for me, it's a personal choice that I want to use the science to try and do something now. If we wait till it's all proven out, it'll be too late for our generation. So, but I clearly know it's a personal choice, and actually you have to measure things. I mentioned that I've been measuring my blood for 10 years or more. I think that's very important to monitor yourself. Otherwise, you don't know if you're doing good or doing harm.
Just to reinforce that comment on combinations, we've been looking at several combinations in animals, and it's entirely unpredictable. So I get really nervous when people are taking 14 different things, because I don't even know what two can do in a mouse together. So I think that's an area we have to do a lot more research in. Max, I want to get to as many questions as possible.
Sure, I think they're enough. We've got a question from David, and he asks, it is generally accepted that chronological age begins when you are born. So when does biological age begin, or biological aging?
Yeah, well, the epigenetic clock begins at fertilization. In fact, it accelerates. It goes very quickly early in life in the first few years, which is really interesting. It means we're aging at conception, at least epigenetically. And I think that's an important concept. Just because you don't see the manifestations of the aging process when you're young, doesn't mean that it's not happening. And if you don't do the right things in your 20s, such as maintain a healthy weight, do exercise, then it will impact how healthy you are in midlife and certainly beyond.
Maybe let's come a little bit to the question of money in the aging field. We've got a question from Liz, who wants to know, how much money do you think we would need to make rapid progress, or even solve the aging problem?
I'll go first, Brian. Solve the aging problem is not really the way to put it. How long is a piece of string? But what I can say right now is that even with the current level of investment, with probably 50 companies now working on aspects of aging, that there will be a company, or many, hopefully, that succeed in having drugs on the market besides the ones that already got on there for other reasons, like metformin and rapamycin. So it's coming, but more money is always good. It accelerates things. If a government could declare aging a medical condition to be treated, that would be one of the biggest things any government could do. or any investment could do. So I'm excited by the billions that are coming in. I think it can only help speed up the drug development and save many hundreds of millions of lives. Actually, every day, over 100,000 people die from what aging causes. And that's very expensive, especially if you get sick early in life. You can spend 10 years being treated. So yeah, I think that all this money coming in is only going to accelerate the research and the development. The amount of money that's coming in, I would say, probably is going to speed it up 5 to 10-fold over what we're currently doing.
And I think that's an important point about governments. It's time for governments to step up and recognize this. I think that, the National Institute of Aging was among, has never been well funded relative to other institutes at NIH, although it was certainly a lot of foresight to start the NIA when the US did. But it's now time that governments recognize that if they want to improve the health of their population, the way to do it, a major way to do it, is to target aging and prevent people from suffering all these difficult problems in old age.
Maybe I combine a question. Jeff Bezos recently has funded a company called Altos Labs, which is working on reprogramming and a few other things. According to your estimation, what do you think, when can we expect Yamanaka factors treatment to be available?
Well, I've been working on this for 5 years now, so we're fairly advanced. We're in non-human primates this month for the reversal of blindness. And then human studies would begin about a year from now, if all that goes well. So it's fairly advanced. If it works in the next few years, we should be able to know if you can reverse blindness. And we reversed the age of human cells. Humans are no different than mouse cells. And we're ticking off the various tissues. So we've done muscle and skin, and we're doing spinal injury now. I don't know about Altos Labs much, other than they have a lot of money and they've recruited most of the field at this point. I'm not going, but
I'm not going.
Yeah. All right, good. Two left standing, but it is exciting that billions are being pumped into that area, and hopefully that money will be used and not squandered. I don't think with that amount of money and with the scientists that are involved, there's a good chance of that. But if there's setup costs, there's setup time. And I think that the companies that are already working on this, there are a few of them, have the greatest chance of being the first to show if this works in people.
Well, we've got a few other questions which deal a little bit with gender. One, do you see any gender differences when you do any supplementations or test those molecules? And second, what do you think about hormone replacement therapy, especially for women?
I'll go quickly because I know Brian has a lot of stuff on this. I don't know of a supplement or a drug or any intervention, genetic or otherwise, that doesn't have a gender bias. There's always one of the genders does better than others, and there are some trends, but it's often really hard to predict how it's going to go. I like to focus on supplements and interventions that work in both sexes, but I think that's one of the big unsolved things in the field, which is what is the underlying basis of this gender difference? And I'll let Brian take the rest of the question.
Yeah, I agree with that. And maybe with the sake of time, what I'll just say is this: we'll get somebody on to talk about hormone replacement therapy, because that's a really common question that comes up. It's clearly on people's mind, and it's a complex issue that I'm maybe not qualified to answer, and certainly can't do in 10 or 15 seconds. So we'll keep that in mind for future shows. David, thanks for coming on. And you have a big paper every 3 months, so can you do the show every 3 months to talk about it? So
that's not true. That old mouse, we made that 10 years ago, and we still haven't published it. But thanks anyway. I've been admiring your career over the last 20 years. So yeah, and thanks for helping me launch mine too.
Oh, it's been fun. And next week, we're going to talk about reprogramming more, because Andrea Meyer will be talking to Menmo Serrano. So I think he'll have a lot of comments on that. He's been working in the field for a long time. Please use the panelists and all attendees button to make comments about the show. We'll try to get to those comments and address them. And we'll leave you with a few thoughts on aging. Thanks again, David.
I'm Jack Hauser, and I want to tell you, a good exercise program is better than any medicine you can ever take. I used to go to a fitness center every day, and I said, well, hey, this is a waste. I bought me a bench. I bought a lot of weights. It cost a little bit over $1 a pound, so that's not much money. You don't have a big investment, but you have a great return. I would do weights, which is strength training, 3 days a week. I usually do 15 to 20 minutes. I walk 2 miles after I do floor exercises. I do push-ups, I do leg lifts, I do sit-ups. They're really amazed that a man at my age can do what I do. To tell the truth about it, I'm amazed myself. The age fitness test is a good way to tell how healthy you really are, and I always tested 10 to 12 years younger than I was. If I can encourage anybody to keep their activity level up, they're going to feel a lot better about life, they're going to feel a lot better about their body, their mind will even be affected by it. I feel like that I'm going to do this forever. As long as I'm here, I want to be healthy and strong, so I will do exercise.
I'm feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.